Okay, moving on to the second part of chapter 6. Let me get my pen ready for you. So, when we left off in part 1, I had told you something about the fact that cell membranes are made up of a lipid bilayer that is composed of what we call phospholipids. So, remember that a phospholipid has a head that is a glycerol slash phosphate group, and it's got a negative charge on it. So this part is soluble in water. And then a phospholipid that's typical in our cells has two lipid chains attached to it, two fatty acid chains. So this part is nonpolar, okay? So here we can see we have the phospholipids all lined up. And you'll notice that some of these phospholipids have kinks in them. We're gonna come back to that just as a review. But the thing that I pointed out to you is number one, that phospholipids, when they're in a water solution, they will spontaneously form a circular membrane, right, that is double layered. So you've got head and two tails, head and two tails, and you can take that all the way around, okay? So now you have a situation where what is inside here is sequestered or shut off from what is outside here if you're in a water solution. Now, if you're a cell, the challenge is going to be letting stuff in that you need, keeping things out that you don't want, but not everything makes it across this membrane super well. So let's review that. Here I have for you things that get across easily on this end of the spectrum and things that have a tougher time crossing that bilayer membrane at the bottom. And what you can see is that if you have small nonpolar molecules, so this is oxygen gas, this is carbon dioxide gas, this is nitrogen gas. These are actually nonpolar because the oxygens, you've got two oxygens, they are equally electron hungry, so they're equally trying to snatch electrons. So the snatching kind of cancels each other out, okay? So these small or nonpolar molecules, what they're able to do is actually slip in between these larger membrane molecules to cross the cell membrane. So getting gases into our cells is no problem. But remember that I told you that this part and this part, they're water soluble, while this interior is nonpolar, right? So you have this nonpolar moat, so to speak, that things have to cross to get from the outside the cell to inside the cell, or vice versa. So you start running into trouble when you get to larger molecules that are polar or just big in size. So let's take a look at water. We know that water is polar, but water is relatively small. So while water doesn't get across as easily as these smaller molecules do, it does get across somewhat. So you can get water back and forth somewhat. Probably not enough to sustain a cell's life, and I'm going to show you in a minute how you can speed up the process of getting water across this membrane. But on its own, you can get some water across this membrane. It's partially permeable. Glycerol is another, it's a 3-carbon molecule, fairly small, and it's nonpolar, so it gets across okay. But again, because it's a little larger, some of it doesn't make it through. All right, sorry, I said glycerol is nonpolar. Glycerol is polar. It's 3 carbons, it's polar. It can make it across okay, but some stuff gets repelled, okay? Glucose and sucrose. Okay, well, glucose, if you remember, it is 6 carbons. I think I went over this in the last lecture. Is that 6? Yep, that's 6. You have a double bond oxygen here, and then on each of these, you have an OH group going across here. Lots of oxygens, right? So we know that glucose is water soluble. We know that it's polar, and we've now gone from 3 carbons in glycerol to 6 in glucose. It's a bigger molecule. Glucose doesn't make it across the cell membrane very well. It's just going to get blocked by this unpolar, nonpolar region on the inside. It just can't do it. So you can see how it's getting blocked here. Maybe 1 or 2 are getting through, but you see how this repel arrow is bigger, smaller here and bigger here. So you can see this repel arrow gets bigger as we go down. So that's going to present a problem. We know cells need glucose, but it can't get across on its own. The very last thing that I want to talk about in terms of permeability of cell membranes is ions. Sodium ions that have a positive charge, chloride ions that have a negative charge, potassium has a positive charge, calcium is another ion that you need that has a positive charge. All of these are things that your cells need, but because they're so highly charged, they're just totally repelled. They're not able to cross this nonpolar region. So they just can't make it through. You can see this repulsion arrow is pretty big. So we're left with the problem, and the problem is that some of the things that cells need can't make it across this membrane. How are we going to address that problem? A second feature of membrane solubility that we talked about a little bit in the last lecture was how many saturated versus unsaturated hydrocarbon tails that you have, or fatty acid tails that you have in your cell membrane. So here you have short unsaturated hydrocarbon tails. That means that this is going to be a more fluid membrane. Here we have longer tails that have saturated hydrocarbons, okay? So this is going to be a more solid or rigid cell membrane. Now, in terms of getting stuff across this membrane, do you think it's easier for things to get across a fluid membrane or for them to get across a rigid membrane? That's going to factor into our membrane permeability, okay? So I'd already tell you here that if it's fluid, things get across easier. It's higher permeable as a membrane. If it's rigid, things have a harder time getting across. You have lower permeability of the membrane. Now let's talk specifically about the processes that get things across membranes. And to understand this, we're going to have to review the concepts of diffusion and osmosis. And hopefully this is going to be review for you. Okay, so let's start with diffusion. And in this case, we don't have any membranes in this scenario. All we have is a beaker, okay, and some dye. Let's just think of this as a drop of dye. You drop the dye in the beaker, and you probably know from experience, and you can see it down here, that if you drop some food coloring or dye, something that's water soluble, in this beaker, over time, it spontaneously will distribute itself throughout this vessel until you have like a constant diluted color of food coloring. So here we have red, and you can see that the red is evenly distributed here. Here we have blue, and you can see that the blue food coloring is starting to become evenly distributed. So why is that? Well, remember that this food coloring is made up of a series of molecules, right? And molecules are constantly in motion. Hopefully you remember that from your high school science classes. They're always in motion. The faster they're moving, the higher the temperature of the solution. But unless you have cooled something down to a temperature that we call absolute zero, molecules, they are moving. So you can almost think of this like you're playing pool, and you've got a bunch of pool balls here. And then as these pool balls start clacking into one another, they start bouncing the individual pool balls around the pool table until they're all kind of dispersed away from one another. So the molecular motion of molecules bouncing into one another pushes all these molecules around until you have them evenly distributed. And what that does is it causes molecules to move from an area of high concentration. So I'm going to say high concentration. Molecules move from an area of high concentration to an area of low concentration. And this happens spontaneously because of molecular motion. Can you see here that this is a high concentration of molecules and this is a low concentration of molecules, that they're more evenly dispersed? So diffusion is what I've just shown you. This is diffusion, okay? And diffusion is just the movement of molecules from a high concentration to a low concentration. It happens spontaneously which means that there's no energy input. I'm going to put NRG input required. So if this were happening in your cell, you wouldn't need any ATP to drive this reaction. All right. Now, let's introduce a membrane. So here I've kind of showed you our little chart that I had earlier. Let's say that we have something that we know moves across membranes very easily. So up here, I have a beaker for you, and we've introduced a membrane, and we have something here that's going to cross this membrane pretty easily. Maybe it's oxygen, okay? And so we have a high concentration of oxygen on this side of the membrane and very little of it on this side. What's going to happen is that through the process of diffusion, we're going to have the oxygen molecules moving over to the other side until we have an even distribution. Let's say that these white things here, maybe they're nitrogen, and they're going to diffuse the other direction. So if these things can move unimpeded across the membrane, then just through the spontaneous action of molecular motion, we're going to have diffusion across the membrane until we reach something called equilibrium, where we have molecules, they're still moving, but they're just, the general case is that the concentration is even on either side, okay? And again, this is diffusion, and this is happening with no energy required, okay? Without any energy input, things will move from high to low concentration as long as they can cross this cell membrane. Now, what happens when we get down here, okay? Especially when you have something like maybe water or glycerol or something like that, that can cross the cell membrane, or that can't cross the cell membrane so well. So, or let's look at glucose. Let's take glucose as an example. It can't cross the cell membrane so well. So what is that going to look like? All right. So in this example, what I have for you is a membrane, just like we had before, but what we have is a high concentration on this side of a substance and a low concentration on this side of the substance. So for the sake of argument, let's say that this is something like glucose that we know isn't going to move across this membrane very well. What's going to happen? Well, you know what does move across the membrane is water. So if the glucose can't move, the water can move. And what will happen is the water will move until, again, we have an equalization of concentrations. So the water is going to move in this direction to equalize this concentration. It appears as though what's happening is it's moving against its concentration gradient, but it's not. Water is actually more concentrated here than it is here. The glucose is more concentrated here than here, but water is more concentrated on this side than this side. If that confuses you, stop the video and try to work that out. So when we have a situation where water is the only thing that can move, but the solutes, the things that are dissolved in the water, can't move, that's a special case of diffusion called osmosis. So osmosis is just the diffusion of water. We aren't violating any of our diffusion principles, right? Because even though there's more glucose on this side than there is on this side, there's actually more water on this side than there is on this side because the glucose here is occupying space. So all that's happened is water has moved from an area of high concentration to low concentration, but the result is that it dilutes out that glucose, okay? So what does this mean if you're a cell? Let's take a look at this. You've probably seen this kind of thing before. So let's say that we have a cell here, or a cell membrane here, okay? And we have a whole bunch of things dissolved in the water surrounding this membrane, okay? These dissolved things are called solutes, and the water is the solvent. On the interior of this membrane, we have a much lower solute concentration. So what's going to happen is the water in here is going to try to equalize things out. We're going to lose water as it tries to even out the concentrations, okay? And so what that's going to do is it's going to cause a shriveling. So if you placed, say, a blood cell into a solution that was full of salt, then it would shrivel, wouldn't it? Now, the vocabulary that you need to know for this situation is what we call tonicity. So in this scenario, we say that the outside solution is hypertonic to the inside solution. So hyper means ramped up, right? If you're hyperactive, then you're super active. So the outside solution is hypertonic. It's super concentrated relative to the inside solution, and that caused water to flow out of the cell and shrinkage to result. Let's look at the opposite scenario. In the opposite scenario here, we have a solution of just really dilute, maybe just a little water with a little bit of salt, or maybe water with a little bit of sugar in it. But inside this cell, you have a high concentration of salt or sugar or something like that. So what's going to happen? Well, water is going to try to rush in at that point to equalize the concentration between the interior and exterior of the cell, right? And if these differences are great enough, that can result in the cell bursting, okay? So in this case, we say that the solution outside this cell is hypotonic to the inside. Hypo meaning slow or less than, right? If you have a hypoactive thyroid, you don't make enough thyroid hormone and you're tired and lethargic. So this is hypotonic relative to the interior of the cell. Now let's move on to what I call the Goldilocks situation. In the Goldilocks situation, everything is just right. You have equal concentration of solutes on the exterior and the interior here. And so molecule or water is actually moving back and forth, but it's doing it at an equivalent rate. So we don't notice any net change between these two situations, right? Before and after. So in this case, what we're going to say is that the outside solution is isotonic, iso meaning equal, isotonic to the inside solution, okay? So this might be important if you're interested in medicine and you were going to give someone an IV, right? You were going to put fluid into their bloodstream. You would really need to pay attention to the tonicity of your IV fluid. You don't want it to be too concentrated, because if it's too concentrated, you could shrivel cells, but you don't want it to be too low of a concentration either. You don't want it to be too dilute, because if your IV fluid was too dilute, what would happen to your patient's blood cells? So instead, when you're putting an IV fluid into a patient's veins, we use something called, usually it's something called lactated Ringer's solution. And that is solution that has been specially formulated to have the appropriate amount of solutes so that you don't end up with a problem over here, okay? You end up with this situation where there's no net change. All right, moving on. Now, we have things that we might want to get into the cell, but that can't cross the cell membrane, things like glucose or ions, okay? So how are we going to do that? Well, we can actually embed some things into the membrane that can function as a tunnel, if you will, to get things from the outside to the inside of the cell. And the things that we're going to use are going to be proteins. So we can insert proteins into this cell membrane that can act as a tunnel. Now, why is it that that works? Well, remember that we have all those different amino acids that we talked about, and the different amino acids have different chemical characteristics. So we had some amino acids that were polar and some amino acids that were nonpolar. So if you have a protein that's constructed in such a way that there are nonpolar amino acids at the ends sorry, I said that wrong, that there are polar amino acids at the ends. Let me not confuse everyone. You want your polar amino acids at the ends with these polar molecules here and these polar molecules here so that they can face the polar water on the outside and the polar water on the inside, right? And then on the interior, you have your non-polar amino acids, so they're cool hanging out with these non-polar tails here, then everything works beautifully. So just like our phospholipids had a polar and non-polar region, you can have proteins that are both polar and non-polar. And remember that the term for that was amphipathic. Okay, so let's talk a little bit more about inserting some proteins into this membrane to help do some of the work of the membrane. Oops, there we go. All right, maybe you remember from high school something called the fluid mosaic model. Now let me back up and tell you a little bit of history. You know, when we first got really, really good microscopes, we could tell that there were proteins that were associated with cell membranes, but for a while we thought those proteins must exist like embedded on the outside and on the inside. We didn't know that they could actually cross the cell membrane. This was what was called the sandwich model of the cell membrane. But we now know that proteins can have polar ends and non-polar ends in a single protein, and that they're actually what we call transmembrane proteins. Trans meaning crossing, right? Transmembrane proteins. Okay, so you can have a protein, for example, that's got an opening here and an opening here, and it can function as a tunnel to get things across. And we're going to look at some examples of that in a second. All right. You still can have some proteins that are on the outside only, and you can still have some that are on the inside only, and then you have some that are transmembrane. And all of these things, they're hanging out on your cell membrane. Now remember that I said to think of your cell membrane like an oil bubble. And you guys have all had the experience of looking at a soap bubble. And you know that if you look at a soap bubble in the sunshine, the soap is swirling over the surface of that bubble because it's actually always in motion. And that's really a great analogy for a cell membrane. These phospholipids and proteins, they're always moving on the surface of this bubble. They're always in motion. So the membrane model that we use today to describe what a cell membrane looks like is called the fluid mosaic model. So if you think about a mosaic picture, if you looked at a mosaic picture in like a museum, it's usually a picture that's made up of many small pieces, many small tiles or small pieces of paper, right? So the membrane is fluid because things are always swirling about, and it's a mosaic because it's made up of individual little pieces that are always swirling about. Hopefully that makes sense to you. Now, to get across this membrane, I'm going to erase for just a second. Boop, 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 boop. To get across this membrane, there are 3 transport types that I want to talk about, okay? The 3 transport types are diffusion, which we've already talked about. And the last 2 are going to require some help from proteins. Number 2 is something called facilitated diffusion. And number 3 is called active transport. Okay, so let's look at facilitated diffusion first. Hi everyone, sorry about that. I had some problems with my screen share, so I'm having to re-record the second half of this lecture. Okay, so I left you with this slide where I told you a little bit about the fluid mosaic model and transmembrane proteins. I also mentioned that our challenge is going to be getting the things that we need across this phospholipid bilayer so that we can have things moving into the cell interior that we want, right? While still keeping out the things that we don't want. And I told you that there are 3 different mechanisms by which we can transport things across membranes. Diffusion is the first one, and we talked about that. But the problem is that diffusion only works for a very small subset of molecules, those things that are small and those things that are nonpolar that can get across this region in here. But when you have those conditions met, like if it's oxygen gas or nitrogen gas, something like that, things can move from a high concentration to a low concentration. So we say that they move down their concentration gradient. This happens spontaneously, and you don't need energy to make it happen. It's just one of those things that happens as a result of molecular motion. But there's a whole lot of molecules that we need to make our cells function that can't get across this phospholipid bilayer. And so what we're going to have to do is insert some proteins into the cell membrane to allow those molecules to cross. And in doing so, we're going to have 2 different transport mechanisms that use these proteins. One's going to be facilitated diffusion, and one's going to be active transport, okay? Now let's take a look at facilitated diffusion. All right, so first of all, when I say the word facilitate, like in facilitated diffusion, the word facilitate means to help. So in facilitated diffusion, all that you're doing is you're inserting proteins into the cell membrane to help the process of diffusion along for those molecules that are not able to cross the phospholipid membrane on their own, okay? And probably the simplest example of that is in channel proteins. So if I were to simplify this diagram, you'd have your phospholipid bilayer here and here. And you would have this channel protein that's essentially functioning like a tunnel, okay? And what this tunnel does is it creates an opening from the outside of the cell to the inside of the cell that molecules can then move across down their concentration from an area of high concentration to an area of low concentration, okay? That's what we're looking at here. So let's take a look at this protein, and you can probably guess, if you were to zoom in on this protein and look at the amino acids in this area of the protein, those would be non-polar amino acids so that they can interact with the non-polar lipid tails on these phospholipids, okay? So non-polar, non-polar. The amino acids at this end of the protein would of course be polar, so they can interact with water on the outside of the cell or on the inside of the cell, okay? Those are polar amino acids. Now this particular channel protein, I'm going to erase that stuff so we can see. This particular channel protein is designed to let water across. So that means that on the inside, in here where the water is flowing, there must also be some amino acids that are polar so that water can interact, all right? This is called an aquaporin. This particular one in this example is called an aquaporin, or if you want to translate that into normal speak, it's a pore for water, right? Now, the neat thing about channel proteins is that they are specific. So what do I mean by that? I mean that, for example, this channel protein is tailor-made for water. Water can pass through, but not other molecules. So the diameter of the opening and the charge of the amino acids inside are just right to let water across, but not other molecules, all right? The other thing is, again, you're moving from high to low concentration, so there's no energy required. Now, one of the disadvantages of just a regular old channel protein is that as long as it's there and as long as it's open, you can't control the rate of things flowing into the cell, right? They're just going to flow in down their concentration gradient. There's no controlling the rate. So let's look at an example of a modified channel protein where you are able to control the rate. So I'm going to actually skip this slide. I decided not to use this slide in this lecture, and I'm going to draw your attention to the following slide. This is a modified channel protein called a voltage-gated channel. And what's going to happen with this channel protein is it's going to allow us to control the rate at which molecules flow through this channel, okay? So let me simplify this diagram for you. Under normal conditions, you would have a channel protein that's in this phospholipid bilayer, but it's got two little arms on it that function like gates and they're closed. So that is this configuration right here. Let's look at it a little more closely. So you have, you can tell I had to record this, re-record this, let me erase that. So here you can see there's my little flaps holding this closed. Now another interesting feature that I need to draw your attention to is these positive and negative charges here. So in this example, we have a situation where the outside of the cell is positively charged and the inside of the cell is negatively charged. And you may be thinking, well, how is that? Well, it has to do with what ions are outside the cell versus what ions are inside the cell. And under a normal situation, you would have a high concentration of sodium outside the cell with a lower concentration inside the cell. So you would end up with a whole bunch of positives out here. But occasionally some things will happen to your cells that will cause those two situations to switch, where you'll have positive charges here and negative charges there. When that happens, when there is a switch in the polarity of the membrane, okay, where your positive hops down here, then that induces a conformational change in this gate and opens it up. So let's draw the second situation. So where these were closed before, now they're hanging open, and now you can have things move through. So we're able to control when things move through by when the polarity of the cell is changed. But in this example, this is some water, but you can see the water is also following some potassium ions here. We're letting potassium through. And it's still moving from high concentration to low concentration, okay? You see that there's positive charges here. These are positively charged. So we must have a lot of positive, oh, I keep wanting to do Ka, sorry. We have a bunch of positive charges here. Potassium is positively charged. So we have a gradient of positive charges that are high here and a low density of positive charges here. So when we open everything up, out it goes, okay? So we're still moving from high to low concentration, so there's no energy required, okay? Let's look at another example of facilitated diffusion using a little bit different type of protein called a carrier protein, but this protein also controls the rate of entry into the cells. So this carrier protein is one called GLUT1, and it's involved in transporting glucose inside the cell. Now let's review. Glucose is polar and glucose is large. And so it's gonna have a really hard time getting across a phospholipid bilayer. But you need glucose. We've discussed how glucose is an energy source for the cell. So we're going to need to take advantage of some transmembrane proteins in order to shuttle glucose from outside to inside. And one protein that can help us with that is this GLUT1 carrier protein. In this example, let me erase this real quick. What you have is a protein that's configured with a tertiary structure such that it has a little pocket here that is just perfect for a molecule of glucose to nestle inside of. And when that glucose binds to this protein, that induces a conformational change where the top of the protein closes and subsequently the bottom of it opens up. When the bottom opens up, this glucose can leave, and now it's been transported into the cell. This can only happen as fast as this protein can change shape. So it causes you to have a sort of slowed rate of influx, whereas if we just had a channel protein, you would be able to move in really quickly. The other thing is you wouldn't really want to have a channel protein for glucose because it would have to be such a big channel, because glucose is bigger than water, that you might end up with other things getting in besides just glucose, okay? All right, so again, in this example, glucose is moving from high to low concentration, and therefore it's moving down its concentration gradient, and so there's no energy needed. Okay, now let's contrast that with active transport. And that's what we're going to talk about here. So active transport is different than facilitated diffusion because in active transport, you're going against your concentration gradient. Normally you'd be going from high concentration to low concentration, but in active transport, you're going from low to high. So you're going in the opposite direction, and that is going to require energy in the form of ATP, okay? So one classic example of active transport is the sodium-potassium pump. You're pumping against the concentration gradient. So that's the chemical symbol for sodium in its ionic form. That's potassium. The sodium-potassium pump, also called sodium-potassium ATPase. So here's a couple of clues about the name. ATP should tell you you're going to need energy. ACE is what we use for enzymes. So this is a pump that's also sort of falls into an enzyme category. Let's take a look at what happens here. So remember how I told you that in a normal circumstance, the outside of the cell is positive because there's a lot of positive ions on the outside of the cell, and the inside of the cell is negative, okay? So what that means is there's lots of, like you see potassium's here and it's positive, there's probably lots of sodium out here, it's also positive. So if we want to move sodium from inside the cell to outside the cell, then we're going to be going against the concentration gradient from a low concentration to a high concentration, and that's going to require energy. Here's how it works. In its unaltered state, the sodium-potassium pump is oriented so that it's open toward the inside of the cell, and there are 3 little active sites here that are just perfect for 3 individual sodium ions to occupy. And they fit perfectly. We say that these sodium ions, or this enzyme has a high affinity for sodium ions. The ions are really attracted to this region. Okay, so once they've nestled in there, if we want to move these from this low concentration to this high concentration area where there's all these positive charges, okay, then we're going to have to use some energy input in the form of ATP, which is right here. So ATP stands for adenosine triphosphate. There are 3 phosphate groups on here. These phosphate groups look exactly the same as the phosphate group that you've seen previously when we were studying nucleotides in the nucleic acid chapters. It's just that there's 3 of them linked together. And each of these bonds contains a lot of energy. These 3 phosphate bonds contains a lot of energy, so that if we snap one of them off, if we break one of these bonds, it's going to give off energy. So that's exactly what we're going to do. ATP comes along, it's going to bind to the outside of this protein. We're going to snap off one of these phosphate groups. When we break that bond, we're going to give off energy. That energy is going to induce a conformational change that causes this region to close. See, it was open, now it's closed. Another consequence of this conformational change is those 3 active sites go away. So now we have a situation where we're closed here, open here. Sodium is no longer bound to this protein, and so it's just going to float out into this region up here. So now we have moved sodium against its concentration gradient. All right. Now the next thing that's going to happen is this conformational change is actually going to open up two new active sites that are perfect for two potassium ions. Let me go through here. Boom. So these two potassium ions are going to bind to these two active sites. When they do, bada bing, here we go, they're going to induce a conformational change back to the original state. The phosphate pops off, these become loosened, and the whole thing can start over. So the end result is we were able to move these positive charges and these sodium ions against their concentration gradient, but it required the use of some energy in the form of ATP. All right, the last thing that I want to show you is another sort of active transport mechanism. And this involves something called co-transporters, okay? So we can talk about antiporters and symporters. Now I've already shown you an antiporter. Hopefully you can see that this is just a simplified diagram of the sodium-potassium pump, or sodium-potassium ATPase. So you can see that this is using ATP to pump sodium out of the cell, and it's letting two ions of potassium into the cell. It's antiporting because it's transporting sodium out while bringing potassium in. They're going against one another. Now, once we have this large buildup of sodium, okay, we can actually take advantage of the fact that we have this high sodium concentration to drive some electrochemical gradient kind of interactions moving down concentration gradient. So now we have a high concentration of sodium that's been set up for us through the action of this sodium-potassium pump. And now we can let sodium flow down its concentration gradient from high to low through a second protein, okay? And in so doing, that induces a conformational change that allows glucose to come in. So we transport glucose, sort of hitching a ride, piggybacking off of sodium. As it moves through this protein, it allows glucose to move through this protein. These are being transported together. So we're going to call this symport, meaning together. And we're going to call this sort of a secondary active transport, because for it to work, you had to use ATP energy to set up this concentration gradient in the first place. All right. The last thing that I want to mention from this chapter is this nice little table here. And what this summarizes for you are the three membrane transport mechanisms that I went over in this lecture: diffusion, facilitated diffusion, and active transport. Here you can see some small nonpolar or small polar molecules diffusing from the outside to the inside of the cell. Here you can see we've had to take advantage of some transmembrane proteins to get things across. And here you can see we've had to do some pumping action. If you can fill in this table, then you understand these concepts and you will be good to go in terms of understanding for your exam. So good luck in your studying. If you have questions about these concepts, I highly recommend going to the study area under the textbook stuff folder on Blackboard. There are some excellent animations and figures associated with this chapter that can help you out. Good luck, and I'll talk to you soon. Bye-bye.